Hello, everyone. My name is Ananta, and today I'll be talking about temporal lobe epilepsy. So, in order to understand what temporal lobe epilepsy is, we would need to first get into epilepsy and seizures. So, epilepsy is basically a brain disorder which is characterized by repeated um, seizures, and a seizure is an alternation of behavior due to temporary change in electrical functioning of the brain. So, normally, your brain constantly generates tiny electrical impulses in an orderly fashion, which then travel along neurons, which is a network of nerve cells in the brain, and then throughout your entire body by chemical messengers called neurotransmitter. Now, in epilepsy, brain electrical rhythms become imbalanced. So, like, there's an imbalance in the electrical impulses, um, and so that results in an reoccurring seizures. So, patient with seizures, normal electrical pattern is disrupted by sudden or synchronized burst of electrical energy, which affects their consciousness, movement, and/or sensation. So, in my research, I also found out if seizures arise from a specific area of the brain, then the initial symptom of the seizure often reflects the function of that area. So, as we know that the right half, there are two parts of the brain. One is the right half, and the other is the left half. The right half controls the left half of the body, and the left half of the brain controls the right side of the body. So, for example, if a seizure were supposed to start from the right side of the brain in the area that controls movement in the thumb, then the seizure may begin with jerking of left thumb or the left hand. And all that I'm saying, the information can be found at the link that I've provided below. If we now come to the picture on the right, as I said, in order to understand temporal lobe epilepsy, we would first need to understand what epilepsy was. So, temporal lobe epilepsy, as it's in the name, it starts in the temporal lobe, which is mainly responsible for hearing, speech, memory, and emotions. So, moving on to the next slide. So as I said in the previous slide, that temporal lobe epilepsy starts in the temporal lobe area of the brain. There are two types of temporal lobe epilepsy. One is the mesial, which is near the middle temporal lobe epilepsy, which is also in the short form. It's spelled MTLE, and the other one is neocortical or lateral TLE. So, MTLE contributes to about 80% of the temporal lobe seizures that start in MTLE. And seizures start in or near hippocampus, which controls the memory and learning. As we learned in the previous slide, that is what the temporal lobe is responsible for. Neocortical or lateral TLE is the seizures that start in outer section of the temporal lobe. And these type of seizures are very rare and mainly due to genetic causes or um, uh, diseases like tumor or birth defects or blood vessel abnormalities. um the causes that I've listed here, and I found were that in children there are birth traumas, infections such as meningitis, encephalitis, and brain abscess, and also high fever and cognitive abnormalities, which lead to epilepsy. In middle age, it's the head injuries, infections, stimulant drugs such as cocaine and alcohol, as well as medical side effects such as tramadol and antidepressants and amphetamines that uh, cause epilepsy. In elderly people, two main causes that have seen to cause epilepsy were tumor or stroke, though there are many that could be underlying. All the information that I'm saying is can be found in the link provided below. Moving on to etiology, the two primary causes are genetic and environmental. Um, for genetic causes, it could be known or presumed genetic defect or problem associated with epilepsy, or spontaneous change in the genes, not which is not always inherited, or inheritance of abnormal genes. It could also be found in families with history of epilepsy, and for environmental causes, I have listed down brain infections and swelling, as well as tuberculosis, HIV, and viral encephalitis, which have been seen to cause swelling of the brain, leading to epilepsy. On the left, I have put here a uh, a, a chart which describes the main causes that lead to epilepsy, and the greatest one is taken by genetic components. Second, most or highest cause is through stroke, and everything that I'm saying can be found in the links down below. Moving on to epidemiology, the prevalence of epilepsy and seizures is 50 million people worldwide. General population with active epilepsy is 4 to 10 people per 1,000 people. As of 2015, 3.4 million people nationwide suffer from epilepsy, which is a huge number. And the ages of that 3.4 million people nationwide was seen to be 3 million being adults and 470,000 being children. Gender, in my study, I found that female usually have lower incidence of epilepsy and unprovoked seizures than in males. And in males, I found that usually it attributes to males' greater exposure to risk factors for lesional epilepsy, such as brain injury, um, though there wasn't more information that I could find why females do not. So, moving on to the region, I found out that high income countries had 49 per 100,000 people per year with epilepsy, and low to middle income countries had 139 cases per 100,000 people. Um, with epilepsy. So, I believe that low to middle income countries have higher occurrence of epilepsy due to not receiving proper treatment and proper care on time. And epilepsy is one of the diseases that require attention on time and proper treatment. And the information I found is linked down below. So the signs and symptoms, often someone having an epileptic episode collapses or has uncontrolled jerking or shaking, um, stiff muscles, loss of awareness, temporary confusion, or just staring spell. Um, Before getting an epileptic episode, people start experiencing strange sensations such as rising feeling in the tummy, unusual smell or taste, and tingling in arms and legs. And the information was found through the website linked down below. So on the left hand side, I have an image which just shows just what to do and what not to do while someone is having an epileptic shock. First thing to do is to cushion the head and remove the glasses if they're wearing any glasses. Loosen the tight clothes. Turn the patient or the person on their side. Uh, they usually have a fixed attack time. Don't Also not to put anything in the mouth of the person. Or search for an ID that they might be having on them. um, don't drink or don't give them anything to drink, they can choke on it. Also, don't hold them down or anything. Just make them lay um, down on the floor, and offer help is the one main thing anyone should be doing while someone is having an epileptic shock or an episode. So diagnosis, coming on to diagnosis, I found out that MRI scans, which is magnetic resonance imaging, um, uh, gives a neuroimaging modality of choice for TLE. Computed tomography, which is a CT scan, um, has a poor resolution compared to MRI, but it's strong for calcified lesions. And a PET scan, which is positron emission tomography, um, interrectal seizure localization in surgical candidates when MRI is normal. So, if MRI is normal, they then do a PET scan. Um, an EEG scan is an electroencephalography, which indicates in all patients with suspected temporal lobe epilepsy. An image on the right, so I've posted an image on the right, which shows an EEG scan detecting wavelengths in the right and the left hemisphere of the brain. And as we can see in the red section, it shows that the wavelengths are different in the right and the left brain in some areas, and that is pointing out to a possible seizure or an epileptic attack. And the information I found is linked down below. So, the pathophysiology, moving on to the pathophysiology, there are mutations that play a role in epilepsy, which is deletion. A deletion is when there's a change in a DNA sequence by removing one or more nucleotide sequence. An insertion, which is instead of removing, just adding one or more nucleotide. Um, Sequence. And a point mutation is a change in a single base pair of DNA by substitution, deletion, or insertion of a single nitrogenous base. So those are the mutations that play a role in epilepsy. Chromosomal aberrations are the strong, like, phylogenetic dysfunction that play a role in epilepsy. Some submicroscopic genetic defects include defects of neuronal metabolism, network development, or membrane and synaptic signaling. Epilepsy can also be due to increased excitability, which happens due to increased activation of NMDA, which is N-methyl D aspartate receptors, overexpression of adenosine kinase enzyme, as well as glycine overexcitability. On the right, bottom right, I have a picture that explains the seizure type, which are focal, which is the basic start. um, focal, generalized, and unknown, which then lead to the epilepsy types: focal, generalized, combined, generalized, and focal, as well as unknown, which then leads to the epilepsy syndrome. And the causes are listed as structural, genetic, infectious, metabolic, immune, and unknown. I was focusing on genetic, infectious, which could be also be environmental. And all the information can be found in the links I provided below. So, coming on to the biochemistry details, I would like to start by describing the picture on the top left hand corner. The structure that is provided is of adenosine. So, adenosine is an anticonvulsant and seizure terminator in the brain, and overexpression of the adenosine kinase. um, Adenosine kinase is an enzyme that regulates intracellular and extracellular concentration of adenosine. So, overexpression of adenosine kinase contributes to seizure generation due to depletion of extracellular adenosine. An increase in adenosine kinase phosphorylates adenosine into AMP, which is adenosine monophosphate, which then creates a gradient, driving the influx of extracellular adenosine inside of the cell. So then, if you don't have, or if you have less extracellular adenosine, the activation of seizure suppression adenosine A1 receptor is compromised, which triggers the epileptic episodes or seizures. The another cause found is due to glycine being both. So, glycine is both excitatory and inhibitory neurotransmitter, and has excitatory effects when it binds to N-methyl deaspartate, which is NMDA receptor, and excitatory actions cause seizures. So, let me explain more about the glycine binding to NMDA. So, if we take a look at the picture on the left, we can see that in the normal synaptic cleft, we see that the NMDA receptor has only one glycine binding to it, and then it releases the um, transmitters to go to grow across the membrane. But on the right side, which says mutation, we can see that there are um, more or more glycine, which is like two glycine in both of the receptors. So that makes it an excitatory response, which causes seizures. And all the information can be found in the links provided below. So moving on, as nerve transmits an electrical signal, sodium and potassium move in and out of the cell depending on the concentration gradient. So when a signal reaches the end of the nerve, neurotransmitters are released to continue the cell-to-cell communication. While they're continuing, it's called an excitatory system, or stopping the communication would be called an inhibitory, since they're stopping. So if we take a look at the figure on the right, we can understand more. So then we can see when there are too many neuroreceptors released, then there is overactivity of excitatory neurotransmitters, or there could be an underactivity of inhibitory neurotransmitters. And this electrical flow of brain is thrown off, um, causing a seizure on an or an epileptic episode. So, a motor cortex and temporal lobes of brain are more vulnerable to have seizures. so the nerve cells are, and why is it it's so why are motor cortex and temporal lobe of the brain more vulnerable? Because the nerve cells in that area are very sensitive to abnormal situations, which cause abnormal electrical transmission due to low levels of oxygen, and also because of infections. So seizures occur when neurotransmitters such as dopamine send electrical signals through brain at a rate that is way too fast, and that causes seizures. If you look at the image on the right, it basically explains um, how the inhibitory synapse works, and um, how the receptors are being transmitted normally. Um, in times of excitatory synapse, um, there's voltage gated sodium channels, and then depolarization. And in the inhibitory synapse, there's glutamate and GABA that are taking place. And all the information could be found in the link down below. Moving on to the treatments. So for treatment for epilepsy, is uh, medications, diet, surgery, and laser ablation. So in medications, they provide carbamazepine, which blocks ion channels and reduces the amount of sodium that enters the neuron in cases of general seizures or epilepsy. Um, and then there is carbazepine, um, levetiracetamine, tar- sorry, levetiracetam, also lamotrigine and topiramate is most effective in patients with MTLE. If you look at the image on the right bottom side, you can see how I've listed the medications used to prevent or treat epilepsy. So as I said, carbamazepine blocks ion channels, reduces the amount of sodium that can enter the neuron. And then there is ethosuximide, which blocks certain types of calcium ion channels in neurons in absent seizures or epilepsy. And then there's lametrigine, which inhibits voltage dependent sodium channels, um, which then decreases release of excitatory neurotransmitters such as glutamate, aspartate, and all those kind of things. And then there's valproate, which reduces H frequency neuronal firing and sodium dependent action potential, enhances GABA or GABA effect. Fulpamate is last resort medicine because it damages the liver or bone marrow, and it seems to be safer in children with severe seizures. So, fulpamate is the last medicine, or the last stage medicine prescribed. A type of diet that they also go by is ketogenic diet because it's high in fat and low in carbs. Um, Another thing is that surgery, if MRI shows like hippocampus or hippocampal sclerosis in MTLE, or uh, mesial temporal lobe epilepsy, and EEG show seizure activity, um, and surgery may cure seizure. So if it shows in the EEG and MRI, then surgery is indicated, and surgery can cure seizures. Temporal lobectomy is removal of temporal lobe, and a temporal lobectomy is the surgery type of surgery suggested in MTLE. So laser ablation is so when an MRI is used to guide the use of laser directed at scar tissue in temporal lobe region, which are causing seizures. This uses heat to destroy tissue, causing the seizure. And the type of surgery that I was talking about, temporal lobectomy, is shown in on the top right hand corner of how it looks. Also, all the information that I'm talking about could be found in the links that I've provided below. Just a quick fun activity. If you could remember and draw the interaction of NMDA receptor and glycine in the synaptic cleft in a mutation versus in a normal situation, to test out if you remember. And then here's the answer. So NMDA receptor, in a normal situation, one NMDA, there are two NMDA receptor, but glycine only binds to one of them. And in mutation, glycine binds to both of the NMDA receptor, which is called an excitatory um, response, causes a seizure. Thank you for your time.